everyone. Welcome to ICANX Talks, Friday night on Connect the World and Universe. This is volume 93. Today, my name is, next slide, please. My name is Lan Fu, and I am a professor at the Research School of Physics in Australian National University. I am the moderator today. So today is a great pleasure for us before going to our talk, first, again, just to remind everyone that the topic of April is biomaterials and applications. So we have already had 2 talks in the past 2 weeks, and today we have the talk from Professor Rashid Bashir from University of Illinois, Urbana-Champaign, and then we have another 2 great talks coming up in next 2 weeks as well. So tonight we have the talk by Professor Rashid Bashir, and also we have a great panelist, the Professor Nicholas Peppers from the University of Texas at Austin, Professor Jen Dongding from Fudan University, as well as Dr. Jia Hao Chen from University of Notre Dame. So first, please let me introduce our distinguished speaker today, Professor Rashid Bashir from the University of Illinois at Urbana-Champaign. Professor Bashir is currently the Dean of Gringer College of Engineering, the Gringer Distinguished Chair in Engineering and Professor of Bioengineering at the University of Illinois at Urbana-Champaign. He has also been the department head of bioengineering and the director of the Holoniac Micro and Nanotechnology Laboratory at the University of Illinois at Urbana-Champaign. Professor Bashir's research is in developing new diagnostic technologies for precision and personalized medicine, and in 3D biofabrication of cellular systems. He's working at the interface of biology and engineering, aiming to make an impact on grand challenges, infectious disease, sepsis, cancer, and others. He has authored and co-authored over 300 journal papers and has been granted 50 patents. He has won many prestigious awards where I don't have time to list all of them today. He's also a fellow of many, many distinguished societies. So without further ado, let's welcome Professor Rashid Bashir to the floor. He will be really talking about his research in forward engineering of multiple cellular biomachines. Please, Professor Bashir.
Thank you very much, Professor Fu. It's an honor to be here. I want to thank the organizers and the panelists for being here and for inviting me. Let me share my slides. Okay, are you able to see this? Great. Let me also just turn on. Okay. Well, thank you so much. So I look forward to spending this next hour to talk about my research interests in forward engineering of multicellular biomachines. So actually my lab is very interested in using macro, micro, and nano fabrication. We use a range of technologies from PDMS, 3D stereolithography, MEMS, nanofabrication, and other biotechnological approaches to then really look at either tools for precision and personalized medicine, and also the 3D biofabrication and cellular systems. So kind of 2 halves of my lab, but my interests are broad, but they do connect, these 2 areas connect. Today, I will mainly be talking about the left side of the screen on biological soft robotics and forward design of biological systems. I can actually give a whole separate talk on the tools for precision and personalized medicine. So let's just get right into it. So we look at the fields of developmental biology, and of course there's so much still to be learned, and there's a tremendous amount of discovery and what I would say reverse engineering that is happening and that needs to happen. Then of course there is this tremendous field of tissue engineering, and the pioneers and the people who invented the field are right here on the screen, Professor Peppers and other colleagues, of course. But then also, I think now what we have also been thinking about is that how do you start to really kind of forward engineer and think about non-natural functions? So can we use our engineering forward engineering approaches to start really designing living systems, systems that are consisting of cells and biomaterials. So that's really kind of the approach here. And one terminology that we've been using is this M-cells, multicellular engineered living systems. And I have to say, a lot of this work actually, from my perspective, how I got involved in this was through a center that actually I had the pleasure of being a co-PI on. It's an NSF STC that is now actually is ending at this time. It was going on for the last 10 years. It was collaboration with Georgia Tech and MIT. Professor Jimmy Shah from UIUC was our campus lead for the first 5 years of it, and then I became the campus lead for the center. But it was the idea here was to think of cells as the building blocks, and the vision was that could we take, let's say, myocytes, neurons, and endothelial cell as 3 cell types that perform actuation, signal processing, and transport, and think about putting them together to sort of build non-natural systems, to build to build machines. So imagine if you have machines that do various functions that are built all of cells and soft materials. And one thing that we proposed, this is a schematic I pulled that was actually drew like 12 years ago, that could we build let's say a biohybrid machine that has the actuation, has muscle actuators, might have neuromuscular junctions, might have a scaffold that houses all of this. Maybe there is some memory and control. There are neuronal circuits. So could you actually sort of design such a machine from the bottom up, so to speak, or from the top down, more from an engineering perspective? So the goal of this was to, again, get this deeper understanding of how cells cooperate and behave, and also to think about these applications that are still, I would say, yet to be sort of defined, but thinking about how you can develop these biological machines. So I'll also mention another NSF grant that we have here. It's a training grant here in the US called the NRT. And this has now also been this 5th, almost 4th year. I'm a co-PI on that. And here also we took the same kind of an idea, but started to think about mini brains or brain organoids. So can we start developing the next generation of researchers in this space of thinking about neuronal inspired machines and computing? So some of the work I'll talk about is also students that were is by students that were in this training grant. So let's take a step back and think about these multicellular engineered living systems. And, of course, like I said, you have developmental biology, extremely complex, a lot of discoveries still to be had. But if you think about from an engineering perspective, could we be thinking about taking cells, differentiating them, perhaps, and then in this case now, morphogenesis, of course, is a very challenging area. But could we then impose the form, I would say, via fabrication? This is where sort of thinking about silicon or MEMS or 3D bioprinting, and then could we then integrate those cells and the scaffold to come up with the machine, right? Another approach, which is more, of course, of the bottom up emergence and engineering, would be to, again, form embryonic bodies or organoids, and then there is co-differentiation, there is morphogenesis, and that results in that machine, which is, of course, certainly very complicated and still a big grand challenge. And perhaps, to move things forward, maybe there is some interface across these two pathways, thinking about how do you develop, maybe such a system. So this space, we also, I like to think about it as in these 3 domains. There are organoids, of course, and there are microphysiological systems, and then these biomachines. So, organoids, obviously now it's a huge field already. Clusters of cells where you have potentially co-differentiation, and you have this minimalistic organ system, perhaps, that the key features of an organ are recapitulated in these organoids. There's a lot of developmental aspects still there, and there is this emergence, starting to have the emergence of some form and function, right? Microphysiological systems also have multicellular behavior, like multicellular systems. I would put them in the same category. But these, I would say, are things like lab on a chip, using microfluidics and this organ on a chip, right? That's also a huge field in itself. Of course, here we're talking about cells that are contained in a device, right? Within a device, and you can interrogate, the function. There is some matrix inside, and you can form now different types of cells and tissues within a microfluidic device. And then these biomachines, I would say, is this a third, application or a domain of this area, which is not in a microfluidic device. It's perhaps more autonomous or self-contained. The space of biological robotics, which I'll talk about, really the focus of talk today. So that's really, I would say, is in this space where the form is more engineered, and the function is coming from the biological sources, the cells. And then you have some sort of machine action, something like actuation, some conversion of energy from one form to the other. So I'll kind of talk about our work in that space today, right? So if you look at the literature, actually, even a comprehensive search, there's probably, I would say, a handful of, not too many, I mean, maybe 10 to 15 examples, I would say, in the literature of this kind of systems that use cells and scaffolds. So this tissue level scale is actually very interesting to build these structures. Some of these devices here, so for example, this is a PDMS pump, which is coated with cardiomyocytes, one of the earlier papers, certainly, and as the cardiac cells actually contract, these sheets contract, you can see here there's a particle, there's a bead actually that shows movement because the fluid is being moved. It's not actually being pumped in this case, but being moved. So that particle sort of goes back and forth. There were a couple of earlier even examples of these robots that were driven by either frog muscles or other types of cells. There is work here in the middle right here from Kit Parker's group and from George Whiteside's group originally that showed this stingray type of, or this PDMS layer that has cardiac sheets. In the bottom here, this is work from Tahir Saif here from UIUC, his colleague here in mechanical engineering. He's also part of our centers. And he has been sort of thinking about building these swimmers. So what you see here is a PDMS long strip that's been microfabricated, and then they put cardiac cells at this interface, and they can get net motion here in fluid. And actually, most recently, just a couple of years ago, they published, I would say, one of the first papers which had a neuromuscular junction actually integrated, which is one of our goals also, as I'll show you. And it showed some initial evidence that the neurons can drive the muscles to actually get net movement. Then there's been more papers recently also. This is colleagues from Japan in this paper in Science Robotics, which actually showed this more of a macro scale, I would say, actuation in this case of this actuator that is driven by two muscles. The muscles are on both sides, and based on which side of muscle you're actuating, you can actually get this actuator to move. So there is a handful of examples in this case that have come about, and they tend to be the system level design and fabrication with driven by cells. So we wanted to kind of move in this space, coming from my original background, as Professor Pepes and others know, is actually in silicon MEMS and bio nano sensors. But I was very interested in sort of this idea of building devices, but more in biomaterials and cells, and that's how we kind of moved in this space. So we said, okay, let's just start very simple, right? So if we want to build something that walks, so the fact is that even an inchworm and how it walks, it's actually very complicated and very interesting, and it is not that simple to just simply replicate it, but at the same time, you can learn a lot from it and actually get inspiration. And so could we build a biobot, a biological robot that walks or swims, right? So my group sort of took on the charge of the walking part, and Professor Tahir Saif here in our center, he kind of took the charge of the swimming. So I'll talk about this walking, right? So could we even fabricate something, sort of a more formal specification would be something that has repeated pulsing muscle contraction with a directional movement that's generated by asymmetry. And we kind of envisioned, this is also from many years ago, sort of a roadmap even, let's say, could we go in this direction? So we try to envision 10 years ago what we would do, and could we use either cardiomyocyte or skeletal muscles and use light or electric signals and eventually neurons, right? So that was kind of our vision. So if you think about a walking biobot, I mean, something that walks, actually there's a couple of, very sort of, I would say, fundamental design principles that the biological actuator must, of course, exceed all opposing forces, including drag and friction, adhesion between the surface and the legs. There must be adhesion, though. A uniformly small friction would cause slippage. So if you think about how something with two legs would walk, right? So one leg has to stay anchored, the other one has to slip, then the other one has to stay anchored, and the back one has to move forward as the actuator in between them makes them move. But there has to be this adhesion, and there has to be some asymmetry actually in that system. And if you look at the actual, let's say, adhesion points here, there's, if you think about this friction of the back leg and friction of the forward leg, there has to be changes, dynamic changes in the friction at different points in the cycle, essentially for the device to move. So using some of those sort of understanding the design rules, we then brought in 3D printing and biofabrication. So this was some of the earlier work. We actually started out by taking a stereolithography system and modified it to be able to then use it with hydrogels and polymers. And we used mainly PEG and PEG-DA of different molecular weights with a photo initiator, ErgoCure photo initiator. And essentially, we're talking about, in this case, resolutions that are still in the few hundred microns in the X and Y. And the Z resolution is about 100 microns. We also kind of built our own micro stereolithography system. And now, of course, you can build these things very easily. Students are fabricating this for a few $1,000 or less even. But we can also then go down to much smaller feature size. This is some printing of these PEG DMA molecular weight of 1,000 on a glass slide. But a lot of this work was actually reported in these papers. And Vincent Chen, who is now working actually at UCSF, Pinar Zoltuluna actually is a faculty at Notre Dame, and Ritu Raman actually, whose work I'll show a lot. She just started an assistant professor position at MIT. So our first version of this was described here, or is described here, where we essentially fabricated these cantilevers and we created an asymmetry by adding this base within this cantilever structure. We mechanically designed this, of course, using finite element simulations first. We fabricated the cantilever within this stereolithography system, activate the surface, functionalize with collagen. And in this case, we seeded cardiac cells from a chick embryo. So these are primary cells. When you seed these cardiac cells, when you extract them, there's also fibroblasts that come with it in the mixture. And the fibroblasts are the ones that grow and multiply and actually form a sheet that then connects the cardiac cells. And we tried different design dimensions, different thicknesses of these cantilever layers, and essentially use this as our first sort of a demonstration of the design and then the fabrication. So again, thinking about the adhesion forces at the interface between the leg and the surface, and extracting that from measuring the contact area. If you look at the friction force as a function of time, a device, in this case, the supporting leg versus the actuating leg, in this case, this friction force does not surpass the supporting layer. So this structure never moved. But if you can actually make one where the friction force of one leg actually increases compared to the other one as a function of time, then you can get net asymmetry and some net movement. So there's again some basic design rules that we can derive. So here is this is about a 7 to 8 millimeter wide structure. You're looking at a cross section in a petri dish in fluid, and as the cardiac cells contract, this this leg right here, or this cantilever, actuates and essentially causes motion from left to right of the structure. Another design is shown here, where again, the cardiac sheet is here on the underside of this cantilever, and the structure can move from left to right. So we can make these autonomous structures that can move. So then again, how do you of course make them more functional and get more complexity? So we were thinking then about actually trying to control the muscles. So we said, well, so we decided to move to skeletal muscle cells. So we started working with C2C12s, which are mouse skeletal muscle cells. And again, we were sort of inspired in this case by looking at physiology and how a muscle tendon bone complex works. So generally speaking, right, you have, let's say, a bone or something with very high stiffness, right, and a joint in this case is what allows the bending. You have tendons again, which is a stiff, high modulus material, and then you have the muscle that then actuates, right? So you can get in this case, it's movement. So we're kind of looking at this, and Ritu and Carolyn, we thought about this sort of a structure which has this sort of a flexible backbone, has two anchors or legs, call it in this case, but these things would have a higher stiffness. And then if you can grow a muscle strip or a muscle tissue around this and get the muscle to actuate, perhaps we can recapitulate this sort of function. So this basic muscle tendon bone biomechanical unit became something that we really sort of fabricated, characterized, and pushed it forward. Of course, if you actually look at the literature, you'll find this structure has been reported by Chris Chen and others in the past, but in a upside down format to measure the traction, to measure the forces that are generated. So the bending of these cantilevers actually can be used to extract and characterize and measure the forces generated by the muscles and by the cells that form the tissue around these beams. So it's sort of the same structure, but now made and fabricated in a way that can be taken out and that can be autonomous, I would say. So if we did modeling of this, again, if you have a symmetric design and you assume certain interfacial forces and friction between the surface and these legs, you don't really get any really net movement when everything is symmetric, because it'll just keep slipping on both ends and not much will happen. So you need to break that symmetry. And in this case, you can break the symmetry by either making the structure asymmetric, as shown here on the right, or potentially even the muscle asymmetric. But in this case, using a structural asymmetry, you can see this thing now moves to the left. And the arrows simply show the starting point, but you can see here that there is actually net movement in this case to the left. So we also worked on that. So then one of the next things that was done that was to look at this process and see how we can also make different kind of strips or rings, we call them. So again, a computer-aided design of the scaffold, 3D printed, then introduced the cell matrix solution, which is in this case this mixture of C2C12, fibrin, collagen, matrigel. And now if you let that matrix compact, it will actually, this entire matrix will actually compact over time and form this very well-defined, in this case, a muscle strip. So this is a muscle cell, C2C12, mixed in with this matrix. And you can see this is a top view image of this structure. The skeleton, what we're calling, these are the two pillars that are sticking up in this case. And it turns out we can also make this, if you change the mold so that there's no gap in the middle, then you can also form what we call a muscle ring. And then this muscle ring, as I'll show you next, can also be taken out and manipulated and actually added on to other structures. So this is a video over many hours of just showing what's happening with this matrix and how the cells in the matrix come together. And I have to say, there's actually a lot of very interesting science still to be characterized here as to the traction forces and the concentration of the cells, and how and under what conditions these cells come together. This is over 12 hours. And you can see that initially it started out with the matrix, with the cells out here. This is the 3D printed structure here. And over time, the cells come together and start to compact the cell in the matrix and form this strip or a ring, right? So this was one of our, I would say, the second generation. This is a top view. You can see this muscle strip with the well-defined structure is the 3D printed skeleton. We have a beam. The modulus of the actual material properties are the same here, but the dimensions is what results in the K being different, essentially, for the beam. So the beam can bend, and these legs don't bend. And essentially, in this case, then this is now electrically driven. So what you're looking on the right is a top view of these biological robots. We apply electric field across them. Electrodes are not shown in this image, but this is electric field driven. And we can apply the signal at 1 hertz, 2 hertz, 4 hertz. You can see the contraction as the rate as a function of time. So it kind of relates to it. It matches the applied field. And then you can because we know the material properties, we can extract and sort of measure the tension, right? So there's actually a passive tension, and then there is the active tension, which is in response to the electric field. So here is 1 hertz, 2 hertz, 3 hertz, and you can see that essentially the device responds, and the active tension is actually in the range of a few hundred micronewtons, was shown here. The passive tension here was about 1 millinewton essentially to start with, but this is the response to the electric field. We characterize this, of course, made sure that the cells are viable, look at the alignment. So this is one of the rings, and what you're seeing here on the top left here, going sort of at the different parts of the ring, where in this along the length, this was the length part right here. So you can see this is this is along this length. The cells are well aligned. As you start to go around the corner, there is actually cells are also curving essentially along that perimeter. This is the other end here. You can see that if you just do image analysis and look at cell alignment, you can see different alignments of the cells, and look at different cell nuclei markers, myosin markers, and also now what we did in this, we actually these skeletal muscle cells are transfected with channel rhodopsin, so they are also now optogenetic. So before I get to that, actually, let me just show you this. This is a video showing the fabrication of these rings in this case, but they are robust enough that we can actually take them out of the original mold and even move them to another mold, not too long in the air, of course. And then in this case, you can manipulate them enough to now attach them to a different structure or a different machine, or think of modular structures essentially, and be able to do things like that. And I'll show you a neuronal stuff later. So then this is our next generation, where this is light controlled. What you're looking at here is a cross section. What you're watching is a cross sectional video in a petri dish in fluid, and the whitish layer is the skeletal muscle. And started out here where the arrow is, and as you shine light, the structure moves to the left, to the right, and it is essentially the directional motion is coming from the asymmetry of the scaffold, how it's printed. So we print it so that one leg has a bigger anchor point, and essentially it then moves from left to right, and it'll keep walking as you shine light in this case, right? And again, we can characterize the displacements and the speeds. On the bottom right, you can see this electrical versus optical. We did both, and we get very comparable simulations. Very comparable results as you stimulate that. But what's also very interesting, and this is described more in the paper, is that if you exercise these using either electrical or optical, then you actually get a higher performance over time. So you can see the speed is faster for the group that was exercised, essentially, that was stimulated more during a training regimen. The general displacement are, essentially you can think about it as right here is 3 millimeters in 10 to 10 seconds or so. So that's about half a body length in about 10 seconds, essentially. And then, as always, we characterize the actual active and passive tensions, right? Now, if you want to optimize it from an engineering perspective and think about what are the design parameters. So Helson did this work, and he's now actually a postdoc at or a postdoc fellow at UT Austin. So the idea of actually, if you look at the muscle and the force, it's a function of the number of myotubes. The more myotubes, the larger actually the muscle, and would also be the force. The force per myotube, which is a function of the individual myotube length. And then the orientation, certainly the alignment. So you can then further kind of break that down, and it has to do with the cross-sectional area of the actuating muscle, the ratio of live tissue to the necrotic tissue, the stress of the individual myotubes, and the alignment. So now you can actually think about designing and optimizing each one of those parameters to try to increase the net force. So we did some work on that, tried to look at different ways to increase that net force. So one thing, for example, was using myoblasts versus myotubes. So we, of course, are taking these skeletal muscle cells. They are myoblasts. We differentiate them into myotubes, and myotubes are the functional cells, of course. But then, as to what point in time do you seed them on the scaffold, on the mold? So a case where the differentiation from myoblast to myotube is happening on the mold versus a situation where the myoblast to myotube differentiation is happening on a stiffer substrate. So it turns out that is actually a more favorable condition. Differentiating them on a substrate, on a stiffer substrate, actually results at the end longer myotubes. And that also allows us to actually get a higher force per stroke when we seed the myotubes into the matrix, rather than differentiating them within that softer matrix. So this is one way to try to increase the force. And then we also did some more of sort of, I would say, empirical or intuitive mechanical designs, where clearly you want part of this region to be thin so that the diffusion of nutrients can get in and the waste can get out, and you don't have much of a necrotic region. So essentially a couple of hundred microns. So we try to make that thin so that there would be a higher viability of the cells. And essentially a lot of those design rules then ended up in these structures that were larger and were able to now produce, active forces of sort of in the millinewton range. Shown here. This is a function of the days of differentiation, 5, 10, 15, and at different frequencies. And we kind of optimized it now to increase these active forces to be in the millinewton scale. Let me skip through some of the stuff here. And now you can see another device here that's walking from top to down. We also, in this case, changed the structure now. I didn't go into that here, but this is now a more a larger muscle strip ring combination that is fabricated and then moved onto the scaffold. We also worked with Professor Mattia Gazzola here in mechanical science engineering. He joined us a few years ago, and he really expanded the work by doing modeling, a more detailed modeling to try to match with experimental results, but then use that to then design structures to be more optimum, and thinking about the efficiencies of, let's say, the bod displacement versus the pillar displacement, and trying to really characterize the slippage. And so his model here predicts our experimental data, reasonably well in terms of displacement and as a function of time. So this is an image of one of these structures. What you're seeing here, it is actually sitting on sort of a glass petri dish, but you can see a reflection. So it's really, the bottom here is a reflection of this top, and this is the backbone, and this is now this whitish layer is this muscle strip ring that also we can fabricate and manipulate. And I showed this earlier. So again, thinking about adding more complexity. Now, the thing is that if you can characterize this basic building block, then you can build higher order structures, right? You can do more complicated things that are in multiple directions. And so Zhazha Wang is a bioengineering student. She decided to add multiple rings. So there's 2 rings. And now using 2 rings, depending on how the electric field is applied, you can actually get this thing to potentially go straight or turn because the electric field is non-uniform. So the structure can turn. One muscle ring gets more stimulation than the other side. We can also model that working with Matthias' group. But essentially, these higher order structures now can be designed, thinking about if you have that basic building block to be more characterized. This is the most recent stuff that actually is in review, but now you can even add multiple rings. You can go 3 plus minus X. You can go 3 sort of the 0, 120 to 40 degrees directions. So here is a structure with 2 muscle rings. Here is one with 3, muscle strips in this case. The way we fabricate it is a strip, 2 versus 3. And you can get this directional motion essentially where if you now shine light on one side, it will move in that direction. And then if you shine light now on the other muscle, it will go back in the other direction, right? There is some asymmetry here still, which is due to the asymmetry of the muscle strip formation, but essentially you can get the structure to go back and forth. And then here is a one with 3 muscle strip. You shine light here, it will move down here. Let me speed this up. So whichever kind of you can then direct them and control that motion more. Okay, so let me kind of add to it next level of complexity. So we'd like to be, start thinking about neuromuscular integration. So how do you get the muscle to, and that's why we kind of went to the skeletal muscle in the beginning, because the vision is to form then a neuromuscular junction and have it neuronal driven, right? So we're not there yet, I have to tell you already, but we're working towards it. Carolyn did some initial work towards it, and you need to really accelerate this now. In our current students are working on this. The idea here was then, could we then do muscle differentiation, muscle strip formation, and then also add on to it the neuronal part, right? So the way we decided to do this in this initial feasibility was to differentiate mouse motor neurons from mouse embryonic stem cells to motor neurons, and use the same process to actually integrate these embroid bodies within the matrix and then let it compact and form a second ring that would have these neuronal embroid bodies. So that was kind of the idea, that it would have to be something that gets done twice, sort of first the muscle, then add the neurons. So we here some characterization of those embroid bodies with the neuronal cells. Essentially the process now is shown here, where you first do the compaction of the muscle layer, and then you add the EBs and the second matrix, which also has muscles and the matrix, and then let that form. So here you can see there's actually two rings that are formed on this mold. So essentially there's 2 layers here. And showed some initial results that were promising. There's some imaging here. Here you can see some confocal imaging of the layer 1, and then so this is a continuous. There is here are the embroid bodies here in the other layer. You can visually here see that. I mean, optically see this here. These darker circular regions are those embroid bodies. So they're not uniformly distributed now, and we're trying to make this process more controlled. But it showed some initial evidence that there are NMJs forming, because when we do chemical stimulation on the skeleton, this is glutamate. We increase the concentration of glutamate, and as you know, glutamate will actually act on the neurons and not on the muscles. So as you increase the concentration, you can see that there's some local twitching that we were able to measure of the muscle. So what you see here is the displacement here as a function of time of the muscle as you increase the glutamate concentration. And then when you add curare, it actually stops it essentially. It just kills the junction. So that, but what is interesting, of course, if you note carefully here, that the displacement is very small. This is in microns. So we're talking, there was local twitching, and we don't have the global contraction yet that we need, which is in millimeter scale or hundreds of micron scale to actually get net motion. So we're not there yet, but that's what we're working on. And that's kind of certainly one of the next steps for that. Let me mention two other important parts of this that we also evaluated with colleagues. So this is in collaboration with Professor Manu Platt, who is at Georgia Tech. One of the things that we found is that these devices, we fabricate many of them and then we stored them, and we found that there's actually some number of these devices as a function of time were degraded. So that strip was degrading, the muscle strip was degrading after a few weeks. And we just weren't sure exactly why. So one of the hypothesis was, well, maybe the matrix is breaking down. This breakdown of the extracellular matrix proteins within the muscle strip is caused potentially by cell-secreted proteases, so cathepsins and MMPs. And it turns out that Professor Platt had this essay of measuring, cathepsins in that matrix. So we actually then did a study with him and looked at whether this eventual breakdown of the muscle strip, what's causing it potentially, and how could you characterize it and potentially even control it. So we just had one paper out on that. I think there's a lot of opportunities to expand this work. But essentially, we were able to show that we actually characterize the survival fraction as a function of ACA. And as you probably know, so ACA is this inhibitor which prevents the plasmin from becoming in its active form and degrading the fibrin polymers. So as you increase the ACA concentration, you could see that actually the strip was more stable over time. So we were actually able to characterize this over many days and even months. Kept the device and looked at its functionality. We would put it back in the incubator and then take it out and stimulate it over time, and then put it back. And we were able to sort of characterize this lifetime that without the ACA, they would generally start to degrade this here, the actual survival here in 10 days. But we're able to extend some of the device lifetime to 30 and some even much more than that. So I think, this is an interesting direction also to see the degradation of these strips and these machines over time, which could be very interesting from different applications. Maybe you want them to degrade after some time, or maybe you don't want them to degrade. So perhaps, that lifetime can be controlled. And then another thing that we did also was to think about healing. So what happens, of course, in actual physiology is, you have these quiescent satellite cells. They get activated, they turn into myoblasts, and then they sort of are recruited to the site. It's a very complicated process, of course, but essentially, if there's mechanical damage, then this mechanical damage in our muscles is alleviated and is counteracted through these recruitment of undifferentiated satellite cells that then differentiate. So what Ritu did, I think, sort of a intuitive but a really cool experiment where she said, okay, let's actually cause the damage and then introduce something to see if we can heal that. And here, I think there's tremendous work in this space, of course, from Professor Pepys, and many others are, the leaders in the field in this, biomaterials and self-healing. We were just kind of getting into this and looking at that. So we actually cause a physical, damage here, and then we add, sort of just manually add this fibrinogen glue, we call it, which was a cell solution, essentially cell and gel solution, where we add C2C12 myoblasts, fibrinogen, thrombin, and matrigel. And then we also, these were optogenetic, so we also then did a split where we would exercise them also during that, call it reconstitution and healing step. So one, some interesting results, but this here you can actually see, these are large enough that you can actually go physically try to do this. So we would cause a damage in this skeletal muscle ring in this case, and then add the biological glue just locally within that region, and then let those cells differentiate and reconstitute within that matrix, within the ring. And then we characterized it. We actually fluorescently labeled the cells that were added with a different color. So you can see that the cells that were added were these green cells, and then the original muscle tissue here was the red one. And after the 17 days, 10 days to 17 days, you can see that this sort of reconstitutes and tries to connect that tissue that part that was damaged. And now if we looked at the actual performance by optical stimulation, the healthy is here. So the original normalized active tension, the percent, and then the damaged, the functionality dropped to about half, because actually the cross-section was roughly, it wasn't extremely well controlled, but enough practice allowed us to cut the thing by about half. When we add just the glue without the cells, we really didn't see a response increase, but then you add the glue with the cell, you actually see a response increase, not coming back to the original, but certainly increasing significant enough, more than just the one with the glue or the damage. So this, trying to show some healing. So the idea would be that this is what you would want the vasculature or some microfluidic system to bring those cells to the site of damage, essentially, to try to heal if there was a damage in these machines, right? So that's kind of what we demonstrated here. Okay, so kind of switching gears on another application. So was talking with John Rogers, who used to be here at the University of Illinois and then moved to Northwestern some years ago. And he said, well, we were measuring the strain and the motion and actually the forces generated optically at this point. It's really, you basically optically characterize the motion, the mechanical properties, so you can back extract the actual forces. So John, as you know, builds these really cool three-dimensional structures. So we said, hey, John, can we build sort of strain sensors that are in the third dimension, and we will put our muscle rings on it. So his group fabricated this strain sensors that are flat first, but then you shrink the substrate so it goes up in 3 dimension and almost forms like those pillars essentially. But in this case, it's this parylene polyimide sensor composite layer. So we worked with them, and his group designed and fabricated this, and then we added our muscle ring to that structure because we can manipulate that, as I showed earlier. And we were able to then stimulate this and actually measure in this case, electrically measure the active tension. So now there are strain sensors on this leg, and essentially when the muscle contracts, you can get an electrical signal. So the idea here was that now you can think about a platform that has many of this, and you don't have to optically measure them, but rather electrical signals for the response. And we did some preliminary drug study response just to see with acetylcholine increasing concentration and caffeine. This is what you would expect for the muscle response essentially. And this signal now is actually an electrical signal here that is a readout from the strain gauge sensors. Right? So this is another direction that I think could be quite interesting and promising. Okay, so next then, we still have about 10 minutes. I'll keep going. So then we were also interested in doing this with neuronal cells. So this is again Helson's work, as I mentioned earlier. So a similar process of that muscle compaction, turns out you can also do that with neuronal cells. In this case, we had to tune the stiffness because we were trying to, well, the thinking was that the stiffness should be softer, in this case, should be less, and it should match or close to, the neuronal cell stiffness. So long story short, we actually use this fibrinogen, fibrin monomer that then turns into this polymer. The stiffness, I think, is about 0.5 kilopascal, certainly less than 1 kilopascal. And that's the matrix we use then with the neurons, and we differentiated the neurons, as I showed earlier, with the embryonic bodies, but then we would dissociate them and then mix them with this matrix. and then let this cross-link and compact. So this process also works and was reported just a year or two ago, where you can see the original distribution, and then as a function of cell concentration and over time, this is at day 4, you can form this tissue strip that is compacted, and essentially this darker layer here after 4 days with this particular cell concentration form this neuronal tissue, which has those differentiated motor neurons. Now, those processes, as you know, are not 100% efficient at all. So we actually characterize that in the paper, and perhaps 70 to 80% of them are motor neurons, and the rest is other types of cells. But they do then, if you place them on a glass substrate, they would grow processes. These are the neurite extensions. And you can count that and try to characterize it. So based on that neurite length and count, we sort of chose the initial cell density that we would use in this matrix. So we ended up essentially right here using this 10 to 12 e to the 6 cells per ml range that would give us sort of high enough neurite count and also high enough long enough neurite length, essentially. So you can now do this with different shapes. You can do squares. You can actually, as a matter of fact, form these squares and leave a region in between here in the scaffold, so there could be neurite connecting these two larger tissues. You can form toroids in this case that would compact and form this toroid. And one, so this actually just showing that this can also be done with these neuronal rings. You can form them, then pick them up in this case, and transfer them into another layer. In this case, Helson put them on a glass rod, and they would then even grow neurites on this glass rod over time. So think of this as building blocks, right? Okay, let me move along because our time is a little short, sorry. So I'm gonna probably skip a couple of these slides, but one very important part I wanna show this was to show that we actually have electrical connectivity within these neuronal tissues. So what we did was we actually made these MEA electrodes, microfabricated simple electrodes on a glass cover slip, and then connected. This is an 8x8 array, and we're able to measure electrical activity. But what we wanted to do was to show that these are electrically connected across the length or the different dimensions of the tissue. So we took the rod, and what you see here is we put the rod on the MEA such that the sensing area is here, but then the stimulation area is a little bit away. So right here is this region is optically being stimulated using light, because these were also optogenetically transfected cells. So we're stimulating them here, but measuring in a different spot. And the idea was that if we're stimulating there, would we see activity down, on the other end of the rod, call it. So that's what you see here, actually pre-stimulation. This is the firing rate that we were measuring. And then you stimulate, the firing rate goes up, and the firing rate is being measured in the sensing area. You stop the stimulation, the firing rate drops, which means that yes, indeed, these cells across this length are actually connected electrically. You stimulate here and you measure here. And the same thing, this is for the toroid. We stimulated up here, but the measurement was on this side. And you see a similar response where the firing rate goes up in the stimulation region when you in the sensing region when you stimulate elsewhere. So this was actually, again, quite interesting to show that these are indeed connected. You can also use these tissues as a drug screening platform, right? Essentially, so that just shows we use in this case human embryonic stem cell derived neural tissue mimics that we place. So it's a 3D tissue, but it was placed on a 2D electrode, and let the cells attach, and then we can measure using these electrodes the firing rate, and essentially it responds to different chemicals and drugs, as shown here. So let me actually jump through this part, and I'll come back to this maybe if there's questions about stimulation and programming of the neural tissue. But in the last 5 minutes or so, so just very quickly kind of summarize what we have shown here over the years is this sort of a roadmap of walking biological robots and machines, in this case, by electric field and light. We looked at lifetime, healing. We made them larger, two rings and turning. And what we're doing now is this new shapes. We're also looking at interfacing electronics with this, working with Professor John Rogers. And then also one of the goals is certainly to try to integrate neurons into this and see if we can have these neuronal controls, right? And in parallel, I showed this neural tissue compaction work, that becomes sort of the neural processor part of this. And then whether that itself, I didn't get to talk about the sort of the training of those tissues here, but whether that could be, those tissues could be trained and or connected back to the muscle. That's kind of the big long-term vision. I do want to spend a couple of minutes just, so again, thinking about these are the kind of things we did and where we're going in terms of the future. But this is a very, I think, interesting area, very unique area that we developed some short courses in this, lectures in different connected areas. It's interdisciplinary, but of course you need the depth in each of these areas. So we do have some of these modules available, taught by different people from within the center. I was also the associate sort of director for education within the center, so we pulled this together. And also at Illinois here, we actually created a class, a junior level, bioengineering class, where we would do this in a class where we teach the students the basic, sort of let them do the mechanical modeling, the 3D printing, the cell differentiation, and essentially say, okay, what kind of structures do you want to make? So you can then think about different sort of pumps and valves and sort of, these biomechanical structures can be fabricated in a lab essentially now. And Professor Kara McCloskey at UC Merced, that I guess didn't play, but she also translated that at UC Merced in a lab. And Ritu actually then recently, also a year ago, put a book out herself on biofabrication also. There's some important ethical considerations to keep in mind whenever we do this biological work, of course, but also in this case, at what level of complexity does a machine become like a living organism? What features distinguish one from the other? What if they can self-repair, learn, adapt? And I hope you see very carefully, I did not say self-replicate. So, because that is clearly, to me, that's where things get really sort of more troubling. But I think this was really important to have our students and faculty and everyone think about this in this area as you're starting to build with cells. We developed these ethics modules that are on the website. And as a matter of fact, we think that there's an ethical framework that needs to be developed. So we actually recruited Matthew Sample, who is a bioethicist, and he put this paper together with many of us in it, thinking about this, so came up with sort of two groups of societal and ethical questions that one would need to consider as we build more machines with cells to do non-natural functions. And then also thinking about, what would be the kind of commitments or you would make for responsible conduct, responsible research in this area. So, that paper, I thought I was a part of that. That was a very interesting experience to kind of think through this with a group of people. And Matthew Sample was the first author. He put that together in this paper just a couple of years ago. One thing I think I'll just talk about broadly speaking, so I do see, of course, engineering is a translation of, laws of physics and chemistry into practice. So over time, right, there's discovery and then there's forward design. So, obviously that's what engineering comes from, I would say physics and chemistry, right? And then more and more, of course, those concepts have also been, I would say biology has been advancing, not only with tremendous amount of discovery, but also this synthetic biology is that forward design, right? How can you start discovering the design rules to forward design? And that you saw some of those thoughts here starting to kind of move in that direction. But, can we do this at this tissue scale, engineered living systems now? Of course, synthetic biology, there's been tremendous advances at the cell level, subcellular and cell level, and now here, could we move in the tissue scale dimension? And then I think that's actually is a big opportunity for health and medicine, of course, as we know. So there is also a paper that a whole group of us put together on just thinking about design rules. This just came out actually this year. Honor Aiden is a postdoc here. Actually, he's an IGB postdoc, postdoc at the Institute for Genomic Biology here, which is kind of a semi-independent postdoc position. He's here at Illinois, and it has people from all across from many institutions. Ron Wise here was the last author, Tahir, myself, and other colleagues here. But thinking about, so this paper tries to articulate sort of at a high level these design rules that one should start thinking about from an engineering perspective as we think about the design rules for these multicellular engineered living systems. And I would say there's many possibilities. There's many different, I would say, these axes of work. There's in vivo to in vitro. What I talked a lot about today was more of the in vitro type applications, but clearly in vivo, this overlaps with, of course, tissue engineering, but thinking about how you can enhance function potentially by adding new things into the design of so can you create a blood vessel that not only transports but rather pumps also. Different types of cells. I think, if you take a step back and think about it, there's actually so many possibilities if you start thinking about insect cells and plant cells and other amphibian cells. Mammalian cells, actually, what we use here they have a little bit of a limitation that they have to be operating at 37 degrees C and in fluid. But what about other types of cells that exist all around us? And of course, this control idea is another important feature to think about. And you can be thinking about swarms of these devices, make many of them. And again, applications in health and medicine, energy, environment, ag, others, but also very importantly, the thinking about the ethics and societal implications. So with that, I'll stop. Pictures of our group over many years, different pictures. Most recently is the one on the right here. And the one at the bottom left. I think a lot of the people who did this work are in this picture actually, on the bottom left and some in the top right. And again, collaborators from our campus and from our center, which is really, I talked mainly about that today. So I really appreciate it. This wouldn't have happened with lots of collaborations. Thank you. And I'll stop here and go from there.
Thank you very much, Professor Bashir. That's fascinating. Before I go to the panel, I think we've got really very active audience today. They have quite a few questions. I have to ask for them. Sorry. The first question is from Rusum Zhao. Is it possible to co-culture blood vessels with other endothelial multicellular organoids?
That's a great question. I will tell you that actually we have not done any work on that. I think there's other people probably here in the panel that can address that. We really focused mainly on muscles and now more neurons, but there are groups around the world that are working on growing vasculature and endothelial cells, as you know. That is one of, I think, a very important area because transport of nutrients and exchange of waste is critical. As the size of the tissue gets larger, beyond the diffusion limit, more than a couple of 100 microns on any characteristic length, I would say, you clearly need to do that. But that's a very challenging area. Like I said, I won't say too much because we haven't published it, but I think others in the panel can probably comment on that. But yes, that is very important. And I think one of the challenges I know in that field is just the stability of that vasculature. You can form the vasculature. One of the challenges is how long can you keep it stable and control the perfusion across the vasculature layers, across the endothelial cell layers, what kind of cells you use. And yeah, because you have to make them so that they're actually permeable. They can be exchange of the nutrients and the waste across that from the source to the tissue around it, right? So, and that is one of our goals, actually. That was Roger Cam and a few others in the center. That was kind of they were focusing on to try to see how you could integrate the vasculature along with the rest of the tissues.
Thank you. And our second question from Xin Liu. Wonderful talk. I saw you design the multifunctional biorobots with dual rings. Did you try other shape of the rings to realize different motion? And how about the size? Can it be micro scale?
Yeah, that's a great question. So we demonstrated these designs that we did, and we really think that the process is now robust enough, and we really focused in on making sure that this is repeatable, even though there's still variability because of just biology and differentiation rates, differentiation percentages of cells, but still it's robust. That yes, we have used that process now over many years, and we did the strips and the rings. And now I showed toroids. So really what we want to do now is make it sort of available and have students and other researchers use different shapes and get different kind of functions. We were thinking of, because this is of course multicellular, and we want to generate enough force to get some actual work, I would call it. We were thinking of certainly staying in this many millimeters to centimeter scale, and actually trying to go even higher. We're thinking about how to scale it up more rather than going down in the micron scale, because obviously these cells themselves are in a few tens of microns each cell, and of course when the skeletal muscle forms the myotubes, they can be very long, right? They can be actually millimeters and longer. You want them to be longer. The goal is to produce single myotubes that are as long as possible, because that's what we have in our body and in physiology. So we want to actually go the other direction by trying to make them larger.
Yeah, that's interesting. Yes. Okay. So our third question is from Martin. I'm wondering how stable the biorings are during motion, given that rough surface may have a significant amount of friction, or if the media has colloidal particles, how would these muscles behave in particular containing media?
So that's also a great question. We have actually a paper almost being submitted very soon where we actually changed the adhesion on the surface by design. So we actually changed the coatings on the surface and made it hydrophobic, hydrophilic, and increased the friction at the interface, and then looked at the speed of the device. So yes, it's going to be very much a function of the viscosity of the medium, but also the surface that the device is walking on, right? And that can absolutely affect the resulting force that is generated and the resulting work that's done. We have something related, I guess, as it was mentioned, maybe perhaps particles and colloid. We've also been working with a collaborator in Spain. Actually, we had a research scientist who visited us from Spain, from IBEC, and they actually integrated piezoelectric nanotubes within the matrix. So the idea about now thinking about how you might enhance the functionality by adding nanoparticles and nanomaterials within the matrix itself. As a matter of fact, on that front, our friend and colleague Ali Khadim Hosseini has a paper on nanotubes, carbon nanotubes, and other sort of carbon-based materials integrated within the matrix, and they showed that allows for an enhanced performance when you stimulate those constructs electrically. You get higher performance. So yes, I think there's a lot of opportunities in thinking about the media that they're in, and also the composition of the material, and what other particles could be added to perhaps sense, provide some sensing output, or thinking about how to enhance the performance.
Wonderful, thank you. Due to the time limit, we'll now have to move to our panel discussion. Now let me introduce our wonderful panel tonight. The first is the Professor Nicholas Peppers. Professor Peppers is the Cockerell Family Region Chairs in Engineering with joint appointments in the Department of Chemical Engineering, Biomedical Engineering, Pediatrics, Surgery, and molecular pharmaceutics and drug delivery at the University of Texas, Austin. So as you can see from here that Professor Peppers is indeed an international leader in multidisciplinary field, where he opened up many entirely new fields of science, generating multiple products and their subsequent generations and collaborating throughout the world. Professor Peppers is one of the most cited scientists in engineering, engineers with over 157,000 citations in total with an H index of 184. So Professor Peppers has also made great contributions to technology translation and education with numerous achievements where I don't have time to list all of them. It's a great pleasure for us to have Professor Peppers today to join the panel. Our next panelist is Professor Jiandong Ding. Professor Ding is a Changjiang Distinguished Professor of Biomedicals and Polymer Science at Fudan University and Director of State Key Laboratory of Molecular Engineering of Polymers. His research is for tissue regeneration materials, drug delivery carriers, and cell material interactions. Professor Ding is the Executive Associate Editor of the journal Regenerative Biomaterials and also a Fellow of International Union of Societies of Biomaterial Science Engineering. So thanks for joining us today, Professor Ding.
Yeah.
And our next panelist and our ex-challenger is Dr. Jiahao Chen from University of Notre Dame. He is a postdoc associate engineering, electrical engineering department at the University of Notre Dame. Dr. Chen received his PhD in materials engineering and science and engineering from Iowa State University under the supervision of Professor Martin Thiel, our old friend Martin. Before doing research at Notre Dame, he was working Professor Samuel Stubbs' group at Northwestern University. He has published over 30 articles and holds 2 patents. So welcome, Dr. Chen. So as per usual, we'll start our panel discussion from our ex-challenger, Dr. Chen, please.
Okay. Thank you, Dr. Bashir. It's a wonderful talk. I learned a lot. So I have a question that is out of my personal curiosity. So as you know that I used to work for Professor Samuel Stubb at Northwestern. John George's lab was actually just next door. So when I was at and when I was in Professor Stubb's group, I was working on this hydrogel material, and I wanted to make those hydrogel materials functional. And those materials are fantastic. And the biggest problem is that the processability. Those materials are really soft and fragile. I had a huge problem to transfer those materials to like a microelectrode array and MEA. So I was very, very frustrated. So after I left Northwestern, so I joined Notre Dame and I joined a 3D printing group. So I was thinking that a 3D printing could be the solutions that to address these processability issues. So my question here is that I know that your groups is going to the other directions, like you make your materials bigger, the scale is a bit larger. So let's say in terms of like a material perspective, a material science perspective, if we want to make this material, the processability is better, or if we will be able to make this materials like a smaller scale, what is your proposed strategy that you would make this materials like a better processable?
Well, that's a great question. So I think, our approach here has been that the cells or the tissues that we are fabricating, there is of course a biomaterial and a matrix within the tissue. So we create the strips and rings, but then those structures are interfacing with these scaffolds, right? So I think there's 2 types of material design sort of considerations, which again, I would say, I think the world's expert, Professor Peppers and Professor Ding and others are. I think the design criteria for the 2 types of materials that I just talked about are going to be slightly different. In one case, you have these materials that cells are within that, and cells have to be, functional, stay alive. They have to have the interfacing within that matrix as they compact and form the tissue. And then the other one is the scaffold that we were printing separately and then growing this tissue on it, right? So the scaffold also has to be still biocompatible. For us, it's PEG DA and PEG DA with MA. And there we want to be able to certainly control the stiffness and the geometry much more so to be able to get that form, I would say, and the resulting performance that we're looking for in terms of, in this case, net force generation or walking. So I think the design criteria are a little bit different, but those are, I mean, I would kind of say those are the two things that we think about. And for both of them, like for the neurons versus muscle, we had to tune the stiffness of that material itself, for example, where the cells are encapsulated in. So I'll kind of stop there and just maybe certainly also welcome feedback from the other panelists there about their advice on this front.
Yeah. So yeah. Okay. The suggested material structure is that we're using a mechanically stronger template or framework, so then we incorporate those functional materials across that frame, right? So it's probably the best solution for this.
Okay. Professor Peppers, you obviously know Professor Bashir's work very well. Do you have any particular questions or comments?
Well, thank you very much, Rashid, for this great talk. I think it makes the students and the faculty reevaluate really how they put together these very intelligent, very unusual M cell structures, and how really we can have someday some organs. I think you and perhaps Aliko Dem-Hasheni are two of the two major leaders in this field. I am a little bit apprehensive, as Dr. Chen is, that others take things and put them together and they don't check the details. Don't forget that Professor Ding and I have done a lot of work in polymers, which you know, and very often we look a little bit with apprehension when we see people putting together things and never checking for unreacted monomer, for toxicity, and so on, and being concerned mostly about the mechanical behavior and so on. I know that your group does that. I appreciate it very much. One question that I have specifically for you, and I know that Victor Ding will ask some things, is how do you decide in your laboratory with your colleagues to take this metal and that polymer and put them together? And granted, sometimes you have to bioadhere, so you put a bioadhesive in between and so on. But how do you decide which material is best to do all these things that you're doing,
Yeah, that's a great question. And I actually didn't talk much about these other very, very important design criteria in terms of materials that you talked about, of course, toxicity and cell adhesion and having bioactive groups within the matrix that attach with, let's say, the RGD groups and others between the materials and the cells. I think those are a critical part of. So we really, in this case, kind of what we want to build off of what's in literature and what's known. But what I would say is the way we think about is not only just the mechanical design, of course, or just the mechanical stiffness part, but rather this aspect that the materials we use have to be cell friendly, right? The cells have to be able to be viable in this and functional and responsive. So most of the materials that we use, which the cells are encapsulated in, end up being right either fibrin based or matrigel based, and materials that are sort of known to be very biocompatible. The other materials that we are using to fabricate with the stereolithography system, they actually have then the photoinitiators, right? And they have to be photo activated to be able to be polymerized and form the three-dimensional structures for the scaffold. And that's why I think this approach perhaps more of an engineering approach, allowed us to separate those two design constraints and be able to then integrate them in a way that we can, get the functional response in a robust manner. So that's kind of what I would. So I think, but what you said is actually critical for the cell, for the I would say more so for the matrix that we are using to form the tissue, that muscle and neuronal tissue. But absolutely, I think those are critical issues to be able to look at the toxicity, the viability of the cells, the necrosis, and we have characterized that in some of the papers. But there's no question we need to go into that deeper and deeper over time. Yeah.
Dr. Fu, I have one more question. I know because of time limitations, we cannot really go into great details. But what I want to say, Rashid, is, and it's really for the benefit of the students and the young scientists, that you, more than anybody else, show something that we call convergence. You bring people from different areas to work on a very important project. So it's not only the electrical engineers and the mechanical engineers, but you have the chemists and the biologists. And the same thing happens right now throughout the world. We see it in China, that I happen to know very well. We see it in Japan, we see it in Europe, and so on. So I am delighted that you're giving, I mean, we met for the first time at Purdue University, as you said, 25 years ago. You knew how to make microcantilevers, I knew how to make hydrogels, and the rest is history. But I do have a last question for you. And the question is really about the whole field. And I know that Victor will follow. It's about the whole field. You hear people like Chris Roy at Georgia Tech and others saying, and Dr. Chen mentioned, they take and they do 3D printing of cells. My God, cells are so sensitive. When I work with them in the laboratory, I try to make sure the pH does not vary, the ionic strength doesn't vary. And here you have people naively saying, I take my cells, I take a prepolymer, mix them, print them, and this is the final material. Is it so easy? That's what I'm asking.
No, it is definitely not easy, and I think that's I think there's no question. There's just a tremendous amount of course detail and finesse in the protocols and recipes and like you said, all those conditions, pH, ionic concentration, cell concentration, and the media type, and especially as you talk about co-cultures. I mean, there's tremendous complexity here with co-cultures and media that would work for neurons versus muscles. I mean, I think nobody has it really that actually figured out yet, I would say. So I think you're absolutely right. And I would say that our approach here has been, if you notice, we're not actually printing any material with cells encapsulated with what I talked about. We did publish on that, but there we went into detail about the viability of those cells, how the products of the photopolymerization, are toxic to the cells, and can we keep the cell viability within that matrix after you do stereolithography, for example. So absolutely, I think printing cells within the matrix and keeping them viable and functional over time is a tremendous challenge, and I don't think it's been as well characterized as it needs to be. So in a way, I think thanks to you and our prior discussions, we really appreciate those problems, and I think we want to certainly make an impact in that, but there's so many people working in that. So what we then decided is to kind of compartmentalize it this way to make progress on this sort of building blocks that I think could be used and replicated and now further really characterized. So I'll tell you one thing. For example, there's really not even a really good characterization of how these cells and matrix comes together to form the compaction of the tissue. So if you really look at the literature, that is not even fully understood as to why and how that's happening and what is the cell. Well, we've done some cell concentration studies and others, but what we've been thinking about doing next is to figure out a way to, label cells and track each cells if we can, each cell and where it's going and how it's interacting in this compaction. So yeah, I think, thank you for the point.
Thank you, and thank you for your answer, and I appreciate it. I don't want to take any more time, Dr. Fu. I think we have also Dr. Ding to talk. So,
yeah. So move to you, Professor Ding.
Yeah. So first, I would like to thank Professor Nicholas Peppers to recommend me to join there as another panelist. So today, I really very enjoyed the talk. It's really a combination of the robotics and tissue engineering, and I have never imagined how beautiful to combine these two different subjects together. So I noticed that today all of the demonstration were given out of body. So have you considered to put one by a robot into one body of an animal and to examine to the real function in vivo, and all how can we imagine the eventual some function in the body or in the future in a human being? Yeah. Thank you.
Yeah, thank you for that question, Dr. Ding. So that's I think that's a critical question. So the short answer is we have not yet put this inside an animal per se. We have designs, and we were thinking of I mean the application we were thinking for an in vivo application would be sort of an optogenetic pump, perhaps, or a stimulation or a ring around another vessel or something to that effect. Or of course, perhaps these optogenetic muscle tendon-like polymer. I mean, a polymer that has the stiffness, much higher stiffness. But thinking about perhaps, replacements or enhancements of optogenetically driven motion or actuation. But we have not done that yet. I think I expect lots of challenges with, of course, degradation from everything that's going to be in the body. And right now, these are still certainly very controlled environment and fluids outside the body. And this is one of the, I think, a very important question that you asked, because if the application, so our goal was and still is thinking about some in vivo applications. But also thinking about how you can create an environment that perhaps has the right enclosure so that the structures could be outside the body. But in that case, you do need to worry about temperature and the matrix and the exchange of nutrients and waste. So this, I guess what I'm coming back to is one of the last slides that I mentioned about the type of cells that one would use. So we did choose to use mammalian cells and work with them in this case because of thinking about applications in the body. And that is clearly one of the next steps, is to start to really look at how they would. And that was our attempt to start to even outside the body right now, the fact is these muscle tissues, and they degrade over time. So this is what Professor Peppers was saying earlier. There is actual degradation because the cells produce matrix and proteins that would degrade the material over time. And I think within the body, these materials, I think, would degrade actually even faster, the ones we're using. So great question. We haven't gone in that direction yet.
Yeah.
We really run out of time, but I do have a last question I want to ask, because I think that's something I really want to ask, is following on Professor Pepper's comments about your field is so encompassing, so broad field, so multidisciplinary, and there's so many young people wants to do that as well. So do you have any very quick advice to young people who are really getting into such a broad multidisciplinary field?
Yeah, that's a great question. And I think, I know we're all very strong proponents of never losing the depth, essentially, in a discipline. I think you have to have So you have to come in from a disciplinary expertise in a certain area. I think focus in on one aspect and really sort of, I would advise to, of course, become really experts in one domain and then bring that domain into a mix where your contributions are very clear as to who is contributing what. I think we don't want to dilute and make the field sort of shallow, obviously, but that is one of the challenges that we all work with in terms of branching out into new areas and new spaces. So you come in from one set of deep expertise and then you expand that. That's the advice I would give. So we don't lose on the depth and the quality of a particular domain. Have the disciplinary depth and build the interdisciplinary breadth. But if you just build the interdisciplinary breadth, that you're gonna lose out of, obviously, on knowing the depth. So that would be my advice.
That's a great advice. Thank you very much. I'd like to thank you, every panelist tonight. That's great talking to you all. And so now, Professor Bashir, I'm representing ICANX. Here I present to you this certificate, virtual certificate, and it will be delivered by email to your mailbox to really show our appreciation from ICANX talk. So thank you for joining us tonight for this wonderful talk.
Thank you very much. My pleasure. My pleasure. Thank you so much. I'm honored.
Next Friday, we have our ICANX talks by Professor Ju Li from MIT. He'll be talking about extreme materials processing for clean energy. So please tune in for next week for this talk.
Thank you. Thank you so much.
And last but not least, yes, I would just like to remind everyone we have ICANX Science Summit 2022 in April 26 to 29. Please join us. We have a list of wonderful speakers going to talk at this summit. So finally, thank you very much for everyone to join the talk tonight. So have a great day or great evening. See you next Friday.